So first of all, I guess we should say, what is dominant, complete dominance versus complete and complete recessive? How would you define that? So think about, the organisms we're talking about are diploid, and so that means they have 2 copies of each gene, 2 alleles. So if we're thinking about complete dominance, how many copies of an allele do they need in order to show the dominant phenotype?
1.
You just need 1. So that's what dominance means. That's what the definition is that you just need 1 allele in order to see the phenotype. And in turn, complete recessive means that you need 2 alleles to see the phenotype. So that's one kind of scenario. And then you have like the incomplete dominance, which means that there's another phenotype, and we'll see that momentarily. Eraser. All right, so if we look at the characteristics of this gene then, and these are cats, by the way. I mean, obviously they're cats, but they're cats that are, oh no, I don't want it like this because, okay, they're cats of different phenotypes. So we're talking here, you can see what is the function of the gene. The FGF5 is the protein made by the FGF5 gene, fibroblastic growth factor. So you can see it is a cell cycle regulator that promotes progression from growth to apoptosis of the hair. Did you guys remember what apoptosis means?
Programmed cell death.
Programmed cell death. So if you had, and there's the way that we're looking at this is cat hair length. So there's long hair cats like this guy. Let's see. We can make this happen. This guy here. So he's a long hair cat, as you might be able to tell. And then like something like this is a short hair cat. Okay, so we're just looking at, that's just a simple complete dominance recessive scenario. So that's why I want to look at it. So if you have a gene that is promoting programmed cell death of the hair in cycles, would you think that the hair would be long or short if it's promoting death?
Long.
Why long?
Aren't hair follicles dead cells or something? I don't remember, but
so in order for the hair to keep growing, then the follicle has to be alive.
okay.
So once the follicle dies, then the hair doesn't grow.
Then it would be longer. Yes. Okay.
Why would it be longer?
Because the cell is alive longer. It doesn't. It's not.
So it's apoptosing, though, right?
Yeah.
Meaning that it's undergoing that programmed cell death, like you said.
Right.
So if it's dying, is the hair going to keep growing or not?
I imagine not.
Not, okay. So it's causing and am I expecting you to remember FGF5 and the function? No. If I do something like this, I will give you a scenario just along the same lines. So apoptosis means cell death. Wow, I never actually used PowerPoint as my writing, so this is interesting. But it was really difficult to put these onto a different program. I'll use that later. Oh my goodness. Okay, it's gonna be problematic. Can't do it. I don't know why it's so delayed. Okay. Well, I'll work on it. Okay, so if the cell is dying, that means the hair is not growing. So it grows for a shorter period of time. So if it's not growing for as long a period of time, then you would expect the hair to not be as long, right? So that means that, and you can see here, I've told you that any protein is enough to make the hair apoptose. So even if you just have a little bit of protein, you promote that cell cycle death. Okay. So what does that mean? That means if you have any copies of the wild type, the functional FGF, the hair dies. So would that be a dominant or recessive characteristic if you if any amount of protein is sufficient. Any amount.
I would say that's dominant.
Dominant. So any amount means that one copy would be enough, which you guys just told me. Oh, and Ariana says dominant. Christina, oh, she's out. Okay, says dominant. Good. So that's the dominant, complete dominant characteristic. Yep. So that's the complete dominant trait there. So what letter would you represent that? We'd do L's, little L's and big L's. So that would be your
Big L.
big L. Yep. And so that means, and you can see this other one, what's the deal here? You have a mutation that is oh, hey, I can write here. Interesting. Can you see the written part that says big L? Did that show up at all on yours?
It was there for a little bit.
It was there for a little bit, and then I stopped sharing, huh? Okay.
I still see where you underline apoptosis and cell death. You wrote that
Yeah.
out. Arrows on the cats, but everything else is not.
It's because I stopped sharing the screen and somehow went to a whiteboard. Share. What?
That was back.
What about now? It's back?
Yeah.
Good. Okay. Yeah, I got you guys back up too. Super. Okay, so that means we got big L, and then little L would be the other one, right? Because that's any protein. So you'd have to have all the protein loss of function. So it's a missense mutation. That means that you have a loss of function of the protein. So without any function of the protein at all, which means you have to have 2 copies of the non-functional, then your hair just keeps growing. So the cat would get long hair. So it's basically this scenario here. And the phenotype is the short hair when the hair dies, and it's the long hair when the hair doesn't die. So you can see how you can connect that genotype phenotype. Now, here's somebody's cat. I don't know who contributed this one, but someone put it on here. Taking a look at this cat, I want you to just I'll take like, I don't know, 30 seconds to figure out or maybe less, the genotype of this cat. What would this cat be genotypically based off of its hair? Remember, it has 2 alleles, so you always know.
2 small L's.
2. Almost. She says, okay, logic for that. People are saying 2 small L's. So 2 little L's mean it has no functional protein for cell death, which means the hair grows long. And indeed, that appears to be a long-haired cat to me. Okay. Yep. So little L. Great. And what about this cat? This is somebody else's cat that they sent in today too. Mason's maybe, or OC.
It could be either big L, little L, or 2 big L's.
Okay, because this is a complete dominant scenario. Good. Then that means that it is you can't tell what the second allele is unless you know something about the parents or babies. It doesn't have to have offspring to be up. Sometimes people think, oh, it has to have offspring to know, but it doesn't have to. You can know the parents as well. So you're correct that this is a big L and then a blank something else. Okay. Let's just go to this, and then we'll do a cross as well in this thing. Okay, so we got a different gene called KIT gene. It makes a receptor that is going to start a signaling pathway. And in the end, that controls the replication and migration of melanocytes. I don't know if you remember from your Genoverse melanocytes, and you might know the word melanin already from, it's just life, right? So melanocytes are going to be those, cyte means cell, cells that produce pigment or Okay, not going to do this in PowerPoint anymore. Okay, pigment. I can't do it. All right. Pigment cells. There's a mutation. It's a large insertion. So this is a different type of mutation, not a missense, but a large insertion, probably from a virus a long time ago. And what that means is that it has a disruption in gene regulation. And remember, it doesn't have to be in the gene itself, but this is gene regulation expression. And so you don't get melanocytes during development. So basically, there's not enough melanocytes to exist for the melanocyte function. So that's one version, that's a mutant version of the gene. And then we have the other version of the gene, which does its normal job. So thinking about this, what phenotype would you think that the mutant cat would have?
Albinism.
Okay, and what would that, so it's only, well, fur-wise. Let's say fur-wise.
Oh.
What kind of fur color does an albino cat have?
Trick question.
White?
Okay,
Yep.
white.
So this is not actually albino. Albino is a different gene. But you are correct in the fact that it's not going to have any color. And so this is what we call, this is a white cat. It's one type of way to get a totally white cat. The other one would be, quote, colored. It would be versions of orange or versions of black. That's how cats are. They're either a version of orange, cream, orange, something like that, a version of black, which can include browns and grays, or a white cat. So this is a white cat. Now, do you think that this allele for the mutant allele would be dominant or recessive, and why? It's a complete dominant scenario again. This would be the dominant or recessive allele. I'm looking at it, and you have to look at the consequence here. So with any mutant allele, specific.
Well, if you're saying with any, it would make it dominant.
Mm-hmm. Yep, that says allele. Wow, this is really bad. We're going to have to go to a different program eventually. Okay, so if it has any mutant allele, it doesn't have enough melanocytes during embryonic development. So you are correct that in this case, the mutant is dominant. And I wanted to put that in there because sometimes people have misconceptions where they think that all mutations are recessive, or they think that all mutations are, cause a lot. Well, this does cause a loss of function, but this is not always true. Dominant and recessive doesn't refer to anything except for whether you need one allele or two alleles to show the phenotype. That's it. Doesn't tell you how the gene's working. It doesn't tell you anything along those lines. So in this case, the mutant is the dominant allele, and we call it dominant white. Actually, this is a different white phenotype than albino, but it's called dominant white. Okay, yep. So my question is, what's the genotype of this cat?
Capital W, or capital W, little w.
Okay, so we can write it capital W blank. I always like to write the blanks so I'm not losing track of what I'm that I need to. Okay, let's tell you now. I'm going to tell you that these are the parents of that same cat. So that cat, now we know the parents of that cat. So you guys told me just from looking at the cat, you know that it's a big W. Now what can you tell me about the second letter?
Heterozygous. So
and that comes from the idea of
them receiving the genes from their parents, whose parents are heterozygous. They're different genes, so they would pass on to their offspring.
So where did this one have to come from?
That one would come from the left cat, or
colored parent.
actually,
So, So we know in order to get color, you have to be recessive, homozygous recessive, right?
Legacy.
So if it's homozygous recessive, then that has to pass a recessive allele to the offspring. So you know that it's a heterozygous offspring. So that's usually what happens is if you know the parent, one of the parents is homozygous, it's homozygous, then you know that that offspring has to have that allele. And why is that? Let's think about this. What is this cat? Let's say this is the female cat. What are her genotypes for her eggs? What kind of genotypes can she have in her eggs? And what can he have in his sperm?
The eggs would probably just have both recessive, and the sperm from the male can have either the recessive or the dominant gene.
Okay, and so what would you actually write for the genotype of the egg?
One uppercase W, and then the little ratio for every one.
I mean, like if I just wanted to say this is an egg, what allele or alleles does it have in it? What's the genotype inside that egg?
Same as the parent.
Okay, so let's look at if it's like this, then we have our egg, and what happens with the egg? Sperm comes and donates the nucleus, right? And so if this was the same as the parent, let's just pretend it's heterozygous. Then what would the baby be?
Oh,
Uh-oh. That's 4 alleles. That's a problem, right? So if we think back, why or how did in animals we produce the eggs and sperm? What process that we learned about this unit?
meiosis.
Which one?
Meiosis.
Meiosis. And so the whole deal with meiosis is it does 2 major things. One thing that it does is generate genetic variation. What's the second thing it does?
It makes 4 haploid cells.
4 cells, and each of those are, what would you call them ploidy wise? You might have said it. Haploid. Okay. So haploid, that means it only has 1 copy of every gene, every chromosome, which is every gene. So that means when you're doing an egg genotype, you will only have 1 copy. So in this case, little w, all her eggs are little w. That's the only egg she makes because she's homozygous for that. And that's why you know that that egg had to make that baby, which is why it has that allele in it. The sperm, we don't know the second allele. We know that some sperm definitely have a big W. Other sperm might have a big W or a little w. It's unclear. While we're at it, how would you know if this white cat had a recessive allele or not? What would you look for in the babies to see? Because this is a test cross where you cross with a homozygous recessive and then an unknown. What are you looking for in the babies to tell you whether it's this or this?
If they have any recessive phenotype babies.
Okay, so if they have any recessive phenotype babies, which would mean that those babies would have to have, in this case, what physical feature phenotype would they be if they were recessive type?
They'd be colored brown or orange.
Okay. So if they have any cats, if they have many litters of cats and they're all white, then you're thinking that it's going to be homozygous dominant. Now, if they only have 2 cats, then that's not enough. But if they have a lot, but if they have cats and they come out, any of them come out with color, that's not white, then you're thinking, yep, heterozygous. So that's what a test cross is. So if I tell you about doing a test cross on the exam, that's what it is. It's crossing with a homozygous recessive, and usually it's to figure out what the genotype is of an unknown complete dominant. Yep, Ariana, yep. Okay, so then this is the same scenario here. Now we'll do a probability. So the probability, which is the fractions of this cat, what's the probability these two parents made this cat? And I want you to do both color, and by color I mean white, not white, based off of the KIT gene, and the hair length. So you're going to have to use both of those to figure out what the probability. And I'll tell you they are unlinked. The genes are on separate chromosomes. So what I would do is to remember the genotypes here. And I'll tell you this cat here is heterozygous for the white. I'll tell you that. It's like, sure.
So are we calculating the probability of the F2 or just the F1?
Calculate the probability that this baby was born.
Oh, okay.
This one. Mm-hmm.
White.
That way they would have gotten a white, short-haired offspring. Okay, so we have some ideas.
I got a quarter, 1 out of 4 chance.
Okay. Let's see what your idea was here. Okay, so we have figured out some of the genotypes already. So what is, let's do the whites then, the Ws. So what do we know already about this? So we'll say this is the mom and this is the dad. So what do we know about the mom cat as far as the KIT gene goes?
So she's heterozygous. So she has
Heterozygous. And well, so why is she heterozygous? I guess we can write this down first. Let's just remind ourselves. L, big W, little W.
Mine.
Okay, so what did L, the dominant here? Long hair. And then the little L had short hair. Gosh, sorry.
Mistake.
This I can't do it. I cannot do it. It's funny because you look at people's suggestions of programs to use for doing lecture and things, and they're like, "Oh, you just write on the PowerPoint," but obviously that is not working for me. Let's, can you see this now? Is this on screen 1 now? No. Sharing. Sketchbook. Okay. So we had that color, the colored long, and then we had the white short, right? Okay. Wow. Something must, maybe it's because it's recording. So it takes a lot of memory and it's not responding. Okay, so we had big L. We decided that was long hair because it just kept growing versus the ones that ate toast, which was a short hair. So to remind us that, we decided that big W was white because there were not enough melanocytes during development, and little w was some sort of color, orange or red based and or black based. So if we have the color cat, we know that that's homozygous recessive for the color versus not color, and then long hair, we just go over here, and that is all we know so far is that it's has one big L. And then for this parent, what do we know? We know the cat's white, so that's big W. I told you it's a heterozygous for white. And then the short hair, we know that this is homozygous recessive. And then we know that the baby, which was the cat that we initially started with, was a white cat with short hair. And so that means it is white. And we don't, so we don't need the W's even because we already know the parents. But what would the second one be anyway?
Little W.
You'll see why we don't actually need that. But and then hair length, the baby has short hair. So then we can go back and fill in this last box here on the parent. So because the baby has short hair, that must mean the long hair parent has to carry a short allele. So it's heterozygous there. And then we just do, what's the probability of getting a white short-haired baby? So we said the only eggs that this parent can produce are little W eggs. And so this is where we do our Punnett square. So there's only one egg option. The sperm option here, there are two sperm options. That's what this Punnett square actually means. And then we say, well, what combinations can you get from those? You'll notice I don't do a second little W because it's like literally doing the same thing twice and you're just wasting your time. Yeah, so we just do it like this. So how many of these options, there's two options, how many of those are white cats, which is what we're looking for here? Just this one in this case. So the probability of getting a white cat is 1/2, and then we'll do the color, and this time it's the same sort of cross, but with the hair alleles, which are for the FGF5. And so which one of these is the short-haired cat?
A
little. So that's out of two options. Now, if both options, like if everything worked, then it would just be 100%, which is 1, which you just multiply by 1, which is this, doesn't change it. So short hair. So what's the probability that you had a cat that's white with short hair? You're right, multiply those together. And so you are, in fact, 1/4 probability. So you can see how you kind of work back and forth sometimes to fill in the genotypes. All right, what's the probability of having the dad's phenotype? You're like, she didn't even tell me what it is, but all you have to do is figure out what phenotype the dad would have based off of the letters, and it's all complete dominance in this case, and then the probability for each of those. So we could do that part together. Probability of dad phenotype. So what does the baby need as A's, B's, C's, and D's to look like the father? So we can do the A, for example. So for A, the A for the father, what type of genotype does the baby need to look like how the father looks? It's complete dominance.
I can't see your screen.
You can't see my screen because I'm sharing PowerPoint. How about now?
Yeah.
Thank you. Okay, so here's my cross. Here's the father over on the left side. So, and we're looking at the baby. We're looking at the A, B, C, and D genes. Each are two alleles, so I've drawn it like this. So for the A, what does the baby need for A's in order to look like how the dad looks like?
Just one, the uppercase.
So if it's complete dominance, any big A will look like the dad. And then you do the B's, the C's, and the D's, and then you just do a Punnett square for each cross. So like for this. And then you see the probability of getting at least one big A. And then you do your B's, your C's, and your D's. So just letting you know not to freak out. You don't have to work through that whole thing. Time. Where did you guys go? There you are. And after a while, you may know the proportions that you expect for these things. Something like this, I would never ask you what's the genotypic ratio of the outcome. That would be crazy. Way too many combos. I would never do that. And we'll do an epistasis next. So do you guys have a number?
I have 3 out of 32.
30 seconds? Okay. I don't actually have this worked, so probably correct though. Big B to look like dad. Big C to look like dad. 2 little D's to look like dad. All right, this cross to get a big A 1/2, because basically it's just, if you got this sperm, then you'd have a big A. If you got that sperm, you wouldn't have a big A. Right, yep, so that's cross there. To get a big B here, oh, it's homozygous. So that means that no matter what, you're getting a big B. And no matter what, you're getting a little b. So period, end of story, every baby is going to be heterozygous, because that's the only options. Yep, so that would be 100% chance. With a dihybrid cross to get one big C, that's the 3. Wow, okay. Would you please not do that? I don't even know how this happened. It's all crooked, right? Oh, there we go. Who knows? All right. Yep. So that's 3/4, because it could be your big C, or big C, little c. And then the little d here, you need one of those, and one of these. So that is, again, 1/2. Yep. So, okay. So across the top is 3. And then across the bottom is 2 times 2 is 4. 4 times 4 is 16. Yeah. Christina, where was yours different?
I think I used I missed that the dad was homozygous recessive, so I used 1/4 instead of 1/2.
For this one, the D?
Yeah, I missed that part.
So you crossed.
Right, 2 heterozygotes.
Okay, so yes, in this case, there's 2 options of eggs and 2 sperm. So to get little d from this cross, you're right, it would be 1/4. But in our case, we have that cross, and in that case, it's 1/2. So this is what I get. Cool. All right, we're going to look at a multiple part thing. All right, found on chromosome 4, this is true. In humans, there's another blood type besides the ABO. There's MN. And instead of writing IA, IB, little I, in this case, we have L, M, and L, N. Didn't copy over quite, but that's, yep, okay. L is this, it's the same gene, the L gene, and there's two versions of it, the M and the N, and they are co-dominant. So heterozygous individuals are said to be in the MN blood group, and that's because they express both of those proteins at the same time on their cells. These are all red blood cell proteins, just like A and B. Okay, so assume a man and a woman, both MN, have a child. What's the probability the child will be a girl and in the MN blood group? So there's two different things there. What's the probability the next two children born to the couple will be in the MM blood group? So similar to ABO, kind of idea with the, but this just has three options: MM, NN, and MN. Okay.
Mariana's saying she's got to go.
Okay. All right. You guys have some ideas for both these? Hi, Ariana. You're welcome. Sorry it took a little bit to get it all together, but we do our best. How are you guys approaching the girl boy?
I mean, that should be 50%, right? That should be half.
Okay, where does that come from?
Well, it's XXXY.
Okay, so this is from the egg and the sperm, and in order to have the daughter, mom always gives an X. It's the only option she has, and then dad can give an X or a Y. So half his sperm are like this, and half his sperm are like that, which is why, I don't know if you remember, but like when they talk about King Henry VIII, he like divorced, or he had 8 wives or whatever, not 8, 6, right? Is that right? Divorced, beheaded, died, divorced, beheaded, survived. Yeah, 6. 6 wives, and beheaded 2 of them because they couldn't give him a son, divorced 2 of them because they couldn't get a son, like started the Church of England because the Catholic Church wouldn't, give him a divorce, and so he just like started his own church, and that's how we have Episcopalians today and Anglicans. Maybe I should, yeah. In any case, so really it's his fault in humans, right? Because he's the one that determines. The females always give. All her eggs have just an X. Now, they have all the other chromosomes as well, but every chromosome for a female has an X. The males have the X to the Y. So, yeah. In birds, though, it's the opposite, where female birds have the heterozygote, or the 2 different chromosomes, and I think it's maybe it's ZZ. I can't remember which one it is, but the males have the same and the females have the opposite. So in that case, the female would determine the sex of the offspring. I can't remember if it's Z's or W's, but that's just You won't need to know that, but just to let you know, it's not always the males in every single case. And then there's stuff like turtles or other reptiles where male female is dependent on temperature. So the eggs that are like closer to the surface are cooler or warmer, probably warmer. So they turn into females in like sea turtles. And then the other ones that are lower would develop into males, cool dudes, and hot mamas. That's how we remember it. I think that's right. So different species have different things. So it's kind of cool. I had a friend that worked in a turtle lab, and basically it's like you incubated the eggs at different temperatures, whether you wanted a female or male for your studies or for your crosses and stuff. So quite different. Okay. So that does mean that, you know, we can do it like this. It's a 50% chance, or 1-2, for both the karyotypes here, both sexes. Okay. So that's always 1/2. So since it specifies girl, then we know that it's half here for this one. And then the MN, so for the second part, we have 2 heterozygotes. And really, it's the same allele. You can do MN, honestly, just to remind you, it's not the M gene and the N gene, it's the M version of the L gene and the N version of the L gene, just to remind you that's not 2 separate ones. It's not 2 separate genes. That's why I like writing it like this. But you could figure it out the other way. So then we want to know the probability of another heterozygote. So this is just like all the other monohybrid crosses. We have big A, little a, big A, little a. Right? So if you work that out, you end up getting this one being mixed combo and this one being the mixed combo right there. And then the other ones being homozygous. Great. So what's the probability of getting the heterozygote? That is going to be this box here would give you 1, and that box here would give you 1. So out of all your options, and you don't have the child yet, so you don't know anything about it, out of all your options, that's going to be half again. And so we take those 2 probabilities and multiply them together. So you would get 1/4. So the probability that you'd have a girl that has the MN blood group is 1/4. That is not the same as this. Not the same. And that's not the same as this. We would never do ratios with fractions. Not a thing. You could say 25%. It's easiest to leave it in fractions just because sometimes you need to multiply them later anyway. So you'll have a calculator on the exam at the top of the window. Okay, what's the probability the next 2 children will be in the MM blood group? All right, good news. We've already done the cross there for the kids. Now it says next 2 children, so that could be either a karyotype, right? So in this case, we don't have to worry about male or female. We're strictly looking at MM. So we have the same exact cross. So we have the same probabilities we had before. So which box is MM blood type? This one here, right? It's the only one that gives us MM. Okay, so that is 1 out of 4, and that's all we have to worry about because it's any kid. What's the probability that their next kid is a girl that is in the MN again? It's the probability they'll have a daughter that's the same as the first daughter.
Be the same, right?
Be the same. So people sometimes, you know, you get confused and think like, oh, hey, Caitlin, we're just working some problems. Yep, it's the same every time. So just because you have, like when you do a genetic disorder presentation, when you say about probabilities for the crosses, you would say, we're on here. You should be able to see the shared screen. Oh. Oh, but you can click on the green. I bet she has to. So do you see next to the space that says review exam 4B, there's like a green button. So click on the green button. You'll see it on your list of things on the side. And I'm coming into this, through Teams, but yeah, so you should be at there. Caitlin's coming in, just that's who I'm talking to. Oh, she can't hear me though, because she's not here yet. That's funny. Click on the green button. Timer. Wow. Okay. All right. Super. Okay. I added this to it. Let's do a sex linked while we're at it. So same scenario here, or not same, but same genes we're talking about. You have the X-linked recessive. That's color blindness. Basically, it's a mutation where your photoreceptors for all the wavelengths don't work the same, and so you can't detect all the wavelengths of light the same that someone who has all functional. So there's 3 different receptors. So if you have different receptors that are non-functional, you get the different versions of color blindness. We're just going to do X-linked recessive red-green color blindness. So it's just a complete dominance, complete recessive.
I'm colorblind.
And then you are?
Yeah.
I try to use colors that are friendly. Or we always do things where it's like, if it's really important, you also bold it or underline it so that it has more than one way to tell people besides being red text. I try to use blue.
Yeah, I didn't find out until I joined the Navy.
Wow. So is it, well, that's a long time to not know. Wow. Yeah. Well, you might want to do your genetic disorder on color blindness. It's complicated, but you're good enough to be able to do it. I think you understand enough to be able to do it. Yeah. Okay. So Caitlin, hello. We're just doing this problem right now about an X-linked inheritance as well as a codominant. So basically we're looking at the MN blood group. So there's 3 different types, just like AB, O kind of thing. But in this case, we call it these types of blood, MM, MN, and NN. These are the 2 alleles. And again, they're just like proteins with carbohydrates on the outside of red blood cells. So I'm combining that with the X-linked recessive. So we have a dad that's colorblind and has this blood type, and a mom who's not colorblind and she has that blood type. And I want to know what's the probability she has a colorblind heterozygous child or a colorblind heterozygous son.
And we don't know if she's heterozygous herself.
Let's see. The mom, not colorblind.
I mean, for colorblindness, yeah.
No colorblind dad. I can say. We'll just go, let's just be a broader.
I don't.
There's no colorblindness in her family. I lost your video, Caitlin. There you are. Okay. No colorblindness in her family. And then let's do one more scenario. So that's one more variation.
I have a question on the first one we did on this page.
Yes.
Would It asked for the next two children, so would you multiply 1/4 by 1/4 or add them?
So if I said, what's the probability that they have 3 children? What's the probability that all 3 of their children have, all 3 of their children are girls with MN? Then you multiply a fourth, a fourth, a fourth, because I'm asking you what's the probability all 3. Now, if I ask you though, what's the probability the next kid is a girl that's colorblind or a girl that's MN, that's the 1/4. So each individual birth, because it's the same eggs and sperm proportions, each individual birth is the same probability. If I ask you what's the probability that 3 or 4 different events happen, then you multiply those together. Does that make sense? That's a good question. Okay. What I can do here. Let's make this happen. Great. All right. So first things first. X-linked means you're having to draw the X's and the Y's and stuff, right? So don't forget that. Y doesn't have anything on the gene. I mean, it doesn't have the same genes as the X, so it's has no allele on it. We have colorblind. It's X-linked recessive, so that means that let's just use A's because C's are difficult, so we'll just use A. So if it's recessive, this means that it's not colorblind itself, but it means that it's the mutant version that leads to colorblind. Say what? And the other one's normal color vision. Or really, it's a functional receptor. So the colorblind dad, we know all males are like this. And actually also it could present this in this way. This means the same thing. So we always know that the males and females have the XY and the XX. Since the father is colorblind, that means he has the recessive allele. Boom, we're done with that. The mother is not colorblind, as she doesn't have color blindness in her family. So she likely has 2 big A's. Okay, what's the probability that they have a colorblind kid? And so you can write this out here. Okay, so how many of those options are colorblind?
0.
None of them.
So unless there's some new mutation, there's no chance that they have a colorblind offspring. So even though the dad's colorblind, they're not worried about a colorblind child. I guess we could say, what's the probability that it makes a carrier daughter?
1.
Ooh.
Oh, I mean 1/2.
100.
Yes, interesting. It depends how I ask it, right? So what's the probability they have a carrier daughter?
Would they both be carriers?
The male can't be a carrier.
No, male can't be a carrier because the male either has it or doesn't because he's hemizygous. So carrier means that it's a recessive allele, or it's a recessive trait that's hidden by the dominant. Basically, it just means that it's completely dominant, completely recessive, and that someone's a heterozygote. So they carry, quote unquote, carry the allele along with them, but they don't show the phenotype. So that's the only way you can be a carrier, is having heterozygous genotype. So you're right, males can't.
I just thought all girls would be the carriers, not males. I know they can because they either express it or not. But when I did my Punnett square, I got, those females would be carriers because the female doesn't have, the mom doesn't have any history, so she's good. But the dad is colorblind, so wouldn't both the females not express it but be carriers? Did I do that wrong?
No. So if I ask the probability of getting a carrier daughter, so there's two things happening here, right? What's the probability? So you don't know anything about the child. So you have two different options for babies. One of those options is the carrier. So the probability that you end up having a baby that's a carrier daughter, because half the time you'll have a normal son, half the time you'll have a carrier daughter. Not you will have, that's the wrong way to say it. You have a 50% chance of having a carrier daughter and a 50% chance of having, in this case, a normal son. But now, so that's if you don't know the baby. Now, if I told you, or ask you, what's the probability your daughter is a carrier, you already know you have a girl. That is out of the equation. So now, what's the probability you have a girl? What's the probability she's a carrier? That's the only option.
100%. Okay, yeah. That's what I was referring to.
So what's the probability that the baby that, so someone's pregnant, that you had the baby that you're going to have, or will be a carrier daughter? Honestly, so it's one of those two options. So that's 1/2. What's the probability that the daughter you've already had is a carrier of 100%? Yep. What's the probability in this one? So this is sort of the same situation here. What's the probability that you have, and we don't even need to do the MN, right? Because we already know that it's no colorblind. And I guess the same thing here, not colorblind for the son either. So in that case, then if we go down to this version of the problem, okay. So now you have an MN dad and an MM mom with she has a colorblind dad. So you don't have to draw the pedigree to figure this out, but just thought it would be nice to look at some pedigrees too. This is also, this is exactly the same thing. So I could represent it in words, or I could give you pedigree. First of all, you couldn't just assume, just because like in this pedigree here, it says both are males that are affected. You can't assume right away, if I don't tell you anything, that that's a X-linked, just because the pedigree is tiny. If I give you a big pedigree, then, and it's all males, kind of getting pretty suspicious that it's going to be an X-linked recessive. But when it's a small one, I could give you something on a test where it's all males, that it's could be autosomal. So what you're looking for here is that, does it look like, are there any kids that have the genotype or the phenotype? Are there any kids who have a phenotype that's different than both their parents? So then we're saying yes, if this is true, and then that means it's recessive. We'll do one in a second. And then we're looking here, and basically you can see that the allele, we're thinking it's passing through the mom, from her dad to the mom. Mom's carrying it, passing it to the son. We want to know what the probability that she passes on the allele is. Okay, so let's look at the dad then. So the dad, he is not colorblind. So what is his genotype? Functional allele. And then let's do the MN as well we're at it. So MN combination. And then the mom, 2 X's. Oh, sorry, that's her dad. These are the mom and dad. Here we go. Okay, so then this mom here, which is this generation, she has a colorblind dad. So her dad here is colorblind, so he has the recessive allele, which means that she must have gotten that from him because she doesn't get the Y, most likely. So she gets the X. And then we know that she is not colorblind, so she must also have a dominant allele there that's functional. So now she is heterozygous, so she's carrier. And what's her blood type, genotype? Oh, MM. 2 of the M's. Okay, so now the probability that she has a colorblind kid, and like I said, you can also just write this out next to it, where you have and so forth, and do your cross like that. So what's the probability of this cross? There are separate ones on the X chromosome, the other one's on the autosome, so you can do 2 separate crosses. So color blindness, we're crossing a carrier cross with not colorblind. This is the most typical passing on of color blindness that you see. So Juan, was your, do you know if your grandparent or your grandfather was colorblind?
You know, Professor, I really never looked into it.
Because you should, because your dad is not.
No, he's not.
No. So then that means it must pass through your mother. And how did she get it? Unless her mother was, her mother could be a carrier. So it could come from her lineage. Or if her dad is colorblind, then she would definitely be a carrier. Interesting.
Now you piqued my interest.
There you go. So is anybody on mom's side a carrier, or was anyone on mom's side colorblind ever? Did anyone hear? Then you know that your grandmother was a carrier that passed it to your mom, and then so forth. But if she says, "Oh no, my dad was colorblind," then you know that it passed through the father's side there.
She's not gonna like that question. I talk to her about biology all the time. She's not.
You don't have to tell her about that it's biology.
I just always bother with what I learned and what I like.
Well, she should feel like she's getting an education for free.
I tell her, Mom, I'm trying to teach you something, Mom. I don't want to learn. Just leave me alone.
It is a good way to figure out if you know things, though. If you can explain it to somebody, that bodes well for your understanding of it. I'm not sure why this is like being so tricky. Let's do this. That's better. Okay. It's awful. There you go. Okay, so what's the probability here of your kid being colorblind? So we're looking colorblind. So how many of these have a colorblind phenotype?
1
out of 4.
Yeah, just this one. Cross with or times your cross here. So mom only has 1 option, dad has 2. Okay, so an MN kid, it's the second box here, right? So you're multiplying those together. So you get an 1/8 chance of having a colorblind kid. That's MN blood type from that cross. What about a son? So then you just go back here and you say, okay, if I'm just looking at the sons, what's the probability?
1/2.
It's 1/2 now instead of 1/4. Same blood cross, so it's still 1/2. And so in that case, if you know, or what's the probability of having the son? So like you know that you're having the son, I should say this. What's the probability, that your son, what's the probability that your son will be colorblind? What's the probability that the son will be colorblind? MN 1/4. So it's higher probability for the boy. So they know they're having a boy. What's the probability that he'll be colorblind? MN 1/4. Okay.
Guess males got the short end of the stick when it came to that, huh?
Well, yes. Now, females, interestingly, there is a Barr body, which you do need to know about that, briefly. So basically, one of the female X chromosomes curls up into what we call a Barr body, and that is not expressed. So that one, it's inaccessible to the expression machinery. So basically, you don't end up getting expression. So, and that makes sense, right? Because if humans are all pretty much the same. So if male humans or XY humans are only getting one dosage of the genes on the X, then females still get one dosage, but that's because one of those genes curls up into the Barr body, one of those X chromosomes, and it's random which one does that or not. And that's actually why you get the cats that are the different colors, where you have the cats that are either calico cats or tortoiseshell cats, depending on how much white they have. Because they have some of their cells that have one X curled up and some that have the other. Let's see if I can. And I would ask you briefly about that. Where? Okay, so cat leg. Must have one on here, like this. So this is a calico version of the cat. Tortoiseshell is like the mixed. I must have a tortoiseshell in. Like that, where it has like some patches of orange, some patches of black, some patches of gray black. This kind of cat, that's tortoiseshell. Calico is the same reason. It just has a lot of white on it. And so basically the deal is here is, we'll just go to this one, each patch of cells, so each chromosome, okay, the gene for the pigment, and this is eumelanin, pheomelanin, same thing actually as the light and dark pigmented mice that we've been talking about all semester. So if there's more eumelanin, it's a black spot. If it's more pheomelanin, it's an orange spot or orange red spot, which is tan, red, something like that, orange. So basically you can see in this cat here, the patches that are black means that the X chromosome, the one that curled up, was the one with the orange allele on it. So that means that the one that's being expresses lots of, makes the cells express a lot of eumelanin, so they're dark. Then in the other, about half of the cells, the one with the allele that codes for black, which means eumelanin, is curled up. So you have more pheomelanin being expressed. And so that gives you kind of an orange patch. And then with the calico cats, like this, same thing here, but you also have another gene that influences how much white is on the cat. So you can see here was a patch of cells. And the reason it's a patch is because one cell in the early embryo gets once it's, I don't know how many cells, not too many cells, early embryo, before it looks like a cat at all, it's just a ball of cells at that point. The chromosome, the X chromosome, inactivates in each cell randomly. And so then those cells keep that inactive and divide. So the one that divided into all this and all this and all of that, and her nose there, that one was one that the orange curled up, and then the other one's the ones the black curled up. So that's how that works. So if you have a tortoise cat or a calico cat, you know it's female because it means it has one orange and one black. Otherwise, if you have two of the eumelanin black alleles, then it's just going to look black no matter which one's curled up. And if you have two of the pheomelanin orange alleles, it's just going to look orange no matter which one's curled up. So it can't be male because males only have one. So they're either orange and that's just expressed, or they're black and that's just expressed. So the only time you'll get, unless it's a Klinefelter cat with XXY, then it has two X's and one will get inactivated. This is also probably why when you did the karyotyping, or we'll do that for tonight, a lot of the disorders, the aneuploidies where they have extra chromosomes, lots of them that are, most of the time, aneuploidy is not compatible with life. So it would either be very early spontaneous abortion or later, but pretty early because it just won't work if you don't have the right dosage of chromosomes. 21, trisomy 21 Down syndrome, can be viable, meaning survive, obviously, because the chromosome is really small with relatively few genes on it. And then the X's and the Y's seem okay because if one X gets inactivated, then if you only have one X in your Turner, you still have that X. So, you're relatively okay. If you're Klinefelter, one of your X's gets inactivated. So it's not like you have a whole lot of extra X being expressed. So it's because of that X inactivation and the fact that Y has very few chromosomes or genes on it, that those sex chromosomes tend to be the ones that we see people being able to develop and be born and live to adulthood with those chromosome abnormal numbers. Really, it's pretty much the sex chromosomes. And 21, a little bit 18, but it's pretty severe. Trisomy 18. 13, even more severe. We're talking like probably not make it to term, but if you make it to term, talking like days, weeks for survival, because just the organs can't do it. They just can't. They can't develop properly without the right amount of gene expression. That's usually why people have miscarriages, aneuploidies. Yeah, and so maybe it helps a little bit. I mean, it's an incredibly emotional event, but to think though that like probably nothing you did. It's probably just because the chromosome number wasn't right and there was nothing you could do about that. That's completely spontaneous when that happens. It has nothing to do with environmental influence or anything along those lines, besides getting old as a female. After a certain age, then spontaneously, eggs tend to have non-disjunction, and that's why there's a lot more Down syndrome offspring to older females. Don't exactly know why, but that's what happens. Let's do an epistasis or two. People tend to be, they struggle on it a little bit more. And I think that's because you get freaked out. You probably know from life experience that there are cats that have patterns and cats that are solid. All cats have a pattern gene. So a tabby cat is the one with like the stripes on it. That's the stripes or swirls, that's tabby. So even if you're a solid cat, you still have genes for tabby pattern. It's just that you don't see those because the solid phenotype means that you don't see the phenotype for the stripes. That's what epistasis is. It means that one phenotype can cover up another phenotype. So it doesn't get rid of the genes. It doesn't even interact with the genes at all. It just, it influences whether you can see it or not. Okay, so here are two examples. These are both black colored cats. So we have the one that's the tabby pattern and we have the one that's solid. In order to see a stripy pattern, first you have to have not solid fur. And the solid fur versus not solid fur is dictated by a gene called agouti. Agouti is the color or the gene like that makes, how like wild animals like squirrels and mice and rabbits and all that have that sort of grayish color. So that's, if you ever look at a hair of an animal like that, you'll notice it has stripes on it, banding patterns. The pigment is expressed, and then it goes through a period where it's not expressed. So it's like a black part, and then an orange part, and then another black part, and an orange part. And so that striping on the hair makes it look like this sort of mottled kind of color. And so you have to have that mottled color to be able to see your stripes. If the hair deposits the pigment continuously, then the cat will look solid. And so you can't see the striping pattern because the solid overrides that, if you will. What that ends up meaning is that we have, write this here. So we have agouti. It's a gene. It's also a phenotype, the agouti gene. So we have two versions of it. There's dominant and recessive. It's complete dominant scenario here. So normally, it deposits pigment and then stops depositing, deposits, stops depositing. And so it makes this striped hair pattern that's dominant. So if it has any of the expression of the agouti protein, it'll make that striped pattern. If it doesn't, if both alleles are broken, it just continuously puts in the pigment and makes the solid color.
Are you putting something on your screen right now?
I am. I'm sorry. I'm sharing PowerPoint. I need to go over to sharing screen number 1. Okay.
All right.
Thank
Good.
you. Yeah, you gotta let me know. Technically can see, but usually I'm not looking at all the windows at one time. Okay, so you've got the agouti gene, codes for agouti protein, I don't know, but actually it's not the agouti gene, it's ASIP, I believe. ASP or ASIP is the gene, codes for protein. And basically it ends up making the hair solid if it's the recessive allele, or if it's the dominant allele pattern. If you have a solid hair, you can't see patterns in the fur. That's the epistasis. There is also a tabby phenotype. There's multiple versions of it. Let's just go with one of them. This is common. There's two, or more than two, but there's two alleles here. They are, the T is dominant to the TB, which is dominant to other T's. This won't do. So let's just consider these two here. So we have one allele that's going to give you the tabby, mackerel tabby, which is the tiger tabby. Okay, and then TB makes like a classic tabby or splotched tabby, we can call it, which is swirly. So it swirls on the body. So those are the patterns. So we're looking at the agouti for stripe versus solid, and then we'll look at the tabby patterns. All right, so you have a mackerel tabby with a solid mom, solid color coat, and a solid black cat with a blotched tabby dad. What's the probability of getting a tabby baby?
A little T.
Interesting. Ideas? See what you want.
Where? Well, you said the mother is solid black, and that's recessive. What does that mean? AB or little AB? Not sure. And then the tabby father, which is big T.
So what you're figuring, you wanted to, okay, yeah, you should probably do the parents first. Is that what you're strategizing?
Yeah, kind of.
Okay, so we know we're looking at 4. Okay. So we have a mackerel tabby. So based off of that, what do we know just from that cat?
Little A.
Well, it has at least one of the T's, right?
So from being mackerel tabby, okay, so to be a tabby and mackerel, we can go here and say that you have to have this one, right?
Yeah.
So at least one T, okay? Now, in order to show a pattern, to even be mackerel at all, to be patterned, what do you have to be a pattern? That's the epistatic part.
At least one of the big A's, the dominant A.
At least one big A. That will give you a pattern. So that we know that. Now, Christina, you said something about a little a coming in. Where are you deriving that from? Uh-oh, I lost her. So the solid mom gives you a clue about that. So if you had a solid mom cat, you know that she was little a.
Yeah.
Which means that the mackerel tabby must be heterozygous for that. And I did this wrong. This should be recessive. Yeah, there we go. Solid black cat. So this is the other one. What do we know about that cat if it's solid black?
That it has 2 small A's.
So that makes it solid. We know nothing else from the cat itself. But it still has tabby genes, even though we don't see it. So I told you that's a blotched tabby dad. So blotched tabby is, you can see here, recessive to mackerel tabby.
So it can
So
either
you must have been
or the
APTB.
heterozygous as well with the big TB or the little TB and the little T by itself. Do we know that?
So I didn't give you a, I'll just do this. There we go. There are other ones, but yeah. So all we know here, we don't know the other allele, but we do know that because he had a blotched tabby dad, blotched tabby it's recessive to mackerel, so it has tissue copies. So we know that that's in play there. Now, the question is, what's the probability of getting a tabby baby? So let's just see what we need for a tabby baby. Even though we're missing some stuff, let's see if we can figure it out. Sometimes people freak out because they're like, I don't know everything, and maybe it's not enough, but let's check. So tabby baby. So how do you get to be patterned, like tabby pattern? What do you need?
At least one of the dominant big T's.
So you need, well, that'll be mackerel tabby.
Oh, I'm sorry.
You could be mackerel tabby, could be blotched tabby.
Oh, yes.
So honestly,
It could be.
any T is going to give you tabby. Now, how do you see tabby, though? How do you see a pattern?
It has to have at least one of the dominant big A's.
You have to have the agouti. Yep, the wild type agouti.
Yeah.
And then the other one can be anything. So in the end, all you need to have a tabby baby, not a solid baby, is a big A. And so we just go and look at our cross for the A's because these are independently assorting. They're on separate chromosomes. So what's the probability of getting a big A? Half, and you can see that here. You're definitely not getting it from the black parent. There's a 1/2 chance of getting it from the macro parent. So that's where this comes from. So really, overall, the chance of having a tabby baby of some sort is 1/2. So even though we didn't know the other information about that, we can still solve the problem. That's why you don't want to go solving for ratios for everything, because it's just like you just want to target what you need to target. Now, what else would I have to tell you? What if I told you that I added those 2 pieces of information now? So now you know that the mom cat for the solid black cat was also blotched. And you know these 2 have already had a litter that had a blotched tabby baby in it. So now I want to know what's the probability. Oh no, let's just do this. What is the phenotypic ratio from this cross here? Expected phenotypic ratio.
Okay. And you said they were both launched. Yeah.
So this is the new information, and this is the new information.
So it means that you can fill in the rest of those, right? Because then
So what's, yeah, so tell me, yeah.
on the left side of the equation, it's going to have 1 TB blotched.
Okay. That's because of the mom or the baby?
That's because of the baby. That's showing you that it's possible to have those 2. I don't know what it's called. The middle dominant.
Yeah, you can call it. It's recessive to tabby. It's a dominant series, honestly, and so each one is dominant to each other. But we'll just consider mackerel and blotch, so it's recessive to mackerel. Yep. So yes. So because it had a blotched baby, then you know that each of them have to have a blotched allele. And how are you figuring out this one on the right?
Well, if the mom is also blotched, that means that she that's another TB.
So both of them have TB, 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 TB. It's just TBs, so this baby must have also gotten TBs. So even though it's a solid black cat, we actually know what tabby pattern alleles it has.
Indeed.
Okay.
And then you want the
separate. Okay, phenotypic ratio. Yeah. Let's just mention, what are the 3? There's 3 different phenotypes that you can have. What kind of cats can you have?
You can have a patterned. Well, with this one, no, I'm sorry. Yeah, you can have a patterned tabby cat.
So we call it a
pattern.
We can have a mackerel tabby cat.
Yeah.
What else can we have?
A pattern blotch tabby.
So you can have a blotch tabby, and we don't even have to call it pattern because in itself, tabby means pattern.
Yeah.
And that's the beauty of it. This is why it's different than saying like 2. Okay, sometimes you look at 2 genes and you're like, okay, it's a pea plant, and you're looking at tall with tall and short and white and purple flowers. So you're looking at 2 things. So in this case, we're actually only looking at 1 phenotype in the end. So it is 2 genes, but in the end, they show 1 phenotype. Yeah. So for example, you have a mackerel tabby phenotype. Yep. You have a blotch tabby phenotype. I'm not saying all these are possible from this cross, but I'm just saying these are options. And then the other kind of cat you could have is a solid cat. We know that these 3 different phenotypes come about through the presence of 2 genes, which each have 2 alleles. That's why I like to draw this out. So basically, you're looking for what A's do they have and what T's do they have. I'll let you take a second. And then what I like to do is just to write my percent or my fractions and multiply them. Like right here. So first I would figure out, what do I need to get a mackerel tabby, a blotch tabby, and a solid cap? Then I do my cross for the agouti, and I do my cross for the tabby, and then I do my probabilities. Then I put it in a ratio.
That's all. And then 4, 10, 5, 10, and half. Okay. And we want a macro tabby. Okay. So I got pretty much across the board, I got halves for everything. So we're just gonna multiply those ratios or these ratios.
Interesting here. Yeah, so you're right that it's the same type of cross 2 times, right?
Yeah.
Because it's a test cross in the end, but it's a heterozygote cross with a homozygous recessive. So in that case, you have both options half the time. Yep. Just to show people watching this. And then it's the same thing again, just with t's. So if you realize that, then you wouldn't even have to draw it again, but you could. And please, please bring a piece of paper for the exam. Anybody watching this? I know it's not, when someone comes into this exam without a piece of paper, I know that's not going to go well. Not boding well for your performance, because like, I can't work these in my head. I don't know. Maybe people are different, but it's not a sign. It's not a sign of weakness to write stuff out. Now, the thing is, though, that you don't want to be finding out stuff you don't need to find out, because it's just using your time unwisely, and you might just get stuck on something needlessly. But so you're right. So we've got half for each of those. So mackerel tabby, we need to know how to get that genotype. So how do we get to be a mackerel tabby? What do we need for our agouti?
One of the dominant A's.
Okay, so we have to have the ability to have a pattern. Yep, big A. Anything else, that would go. And then to be mackerel, we have these options here.
Yeah, one of the dominant T's.
Okay, a big T and then either another big T or a T little b, doesn't matter. So that's how you get to be a mackerel tabby. Hey, Christina, you're back. How do you get to be a blotch tabby?
A blotch tabby, you need 2 recessive TBs. That the T with a B with a little b.
Yes, and you can say little and big. Little and big's okay.
Okay.
I lost my brush pen. Yeah, okay. So to be blotched, you again have to have the ability to have a pattern. And then, wow. To be blotched, you have to have we need a new layer. Wow. Nobody knows. Nobody knows. Why you aren't writing. New brush. Okay. There we go. Cool. All right. And then to be solid,
Need.
yep. That's it. Any pattern that would go. So then you're right, we go and we see which boxes fulfill these criteria and what probability is. So probability of getting a big A is 1/2. And the probability of getting a big T is 1/2. Okay. Probability of getting a big A is the same. Probability of getting T little b, 10. It won't always be the same numbers, so don't trick yourself into that. What's the probability of getting 2 little a's?
1/2.
1/2 again, and the probability of getting any T, like 100%. So that equals 1. And this is good. It should add up to 1 in the end, unless there's some sort of death going on. Okay, so then you turn that into a ratio.
So it would be like.
So it all has to have the same denominator. So we can get that to be. So what do we expect? We expect 2 solid babies for every 1 mackerel tabby baby, for every 1 blotched tabby baby. Will it happen exactly like that? No, but that's the ratio we'd expect, especially if they have a lot of litters. If you have something like fruit flies and you have like hundreds of offspring or thousands or tens of thousands of offspring and you're not seeing these ratios, then you know that something is probably going on where the genes are probably linked. So 2 of the genes are on the same chromosome, which means that they do not do this segregation. The only reason we can work these 2 Punnett squares separate is because the A's do their own thing. They sort themselves through independent assortment. And the T's do their own thing because they're on separate chromosomes that, independent assortment during metaphase 1. And so that's why you can work them separate, because they don't influence each other. But if they're linked, then you're looking at not getting the right ratios that you're expecting. And then you can't do the problems like this. If they were linked, I would never be able to ask you to work the problem. But I could certainly tell you something like the rate, like the proportions of offspring you're seeing, and you can tell that that's not right. And then you could say that those genes are probably linked because they're on the same chromosome.
How do you get the little t superscript b on the left-hand side of the parent, the left parent?
This one here?
Mm-hmm.
So they've already had a litter of babies, and they've had a blotched tabby baby in there. Yeah. You want to go crazy? Add one more layer on this? Okay. And like literally though, everything's just taking each. Now this case, you did have to consider both of these at the same time because it's the epistasis. But I'm going to add another layer on top of this, which is totally separate, but it's the orange versus black cat. And I had mentioned that before, is that you have an orange, which is big O, which produces more pheomelanin. The little o allele produces more eumelanin, which is a dark pigment, so it ends up coding for, it gives you a black phenotype. So those are the 2, and then these are X inactivation, tortie, which is tortoiseshell, and we want it to be mackerel tabby. So I think they actually call it a torbee, a tortoiseshell tabby cat. That's a probability that, now I've told you some other information. So the additional information I've just added here is that this is the female and she's orange. This, we already know it's black, but that's the male. And I want to know the probability of having a tortoiseshell, which remember is that mixed pattern. patches, some black, some orange patches. And then in those in those patches, you see tabby pattern. Oh my, that's on a separate layer. Oh, no. So you can use these probabilities you already figured out, right? And all you need to do is to add on to that the orange-black aspect. How am I going to link these? Oh, that was good. Sometimes I am. I mean, I'm good at technology. There's just a lot here, but just link those layers without even knowing what I was doing. Awesome. Okay. Yes. So this is X-linked, but it even has to do not just with X-linked, but with that X inactivation. So to get an orange female cat. So if she's female, the 2X is happening here. And she's orange, which means based off of this, what alleles does she have?
At least one of the oranges.
Remember, there's this. Because of that X inactivation deal, we have mixed patterns when you have some of the cat being orange and some of the cat being black, or some version of those colors, and sometimes white. And so anytime you have a heterozygote, you're going to have a tortoiseshell or calico pattern. So we're even more certain about what this cat's genotype is. So since she's just plain orange, we know that she has to have another big O. If she had a little o, she'd be tortoiseshell or calico, depending on how much white she had. Which is interesting in this case, so you can tell. And the male is black cat, so he's the little o version, produces more eumelanin. And then I've asked for a tortoiseshell mackerel tabby, or a torby, mackerel torby. Okay, so let's figure out what we need to get that. And let's, I know you can use this here and just multiply with it. Let's just do it out though, like we were doing it from. Make sure. All right, the probability. Okay, how do we get to be tabby pattern? Okay, we have to be patterned. We found out before that to be tabby patterned means that we have to have one big A, or else you're solid. To be mackerel tabby, we have to have the one big T. At least one. And then, in order to be, I'm asking about a tortoiseshell. So what must the tortoiseshell baby be?
Big O, X little o.
X big O, X little o. So first of all, we know what sex the baby has to be in order for this to work. You'd end up with a female baby. So let's do the cross for the X's now. We've already figured out these other ones, and just the same as before, right? You could just use a 1/4, but in this case, we have the big A. We have the big T, at least one. Now, we have a parent with only one option, so the mom only has eggs with the O. The dad has half X's and half Y's. Now, of course, the only one that'll give you a tortie or tortoiseshell is this one. So the probability of that in this case is also a half. 3 times 2 is 4, 4 times 2. Do you agree with me? Okay. So the probability of these of this orange mackerel tabby and the solid black cat having a tortoiseshell baby with a tabby pattern in its patches is 1/8. So you can see, oh my God, she gave me 3 things. Just take it figure out which ones matter together. The agouti and the tabby matter together. The tortie, on the other hand, the orange-black tortoiseshell, that is its own thing.